Hi everyone, and welcome to the 24th PHAVES seminar. I can't believe we've done 24 now. It feels like just yesterday we started. But just a reminder that we have a 25 to 30-minute seminar from Julianne and Whitney, who I will introduce in a moment. But while the seminar is on, you're more than welcome to type questions in the chat and I can read them out at the end. Or of course, there will be a chance for you to unmute during question time, which will follow the seminar. We will then, following the questions, we'll have our breakout rooms, time permitting, where we will just randomly assign you to a breakout room for just some really casual networking for about 5 to 10 minutes. So yeah, that's the layout for today. I just quickly want to say thank you to Phage Futures for sponsoring this seminar today. There is still time to register, and if you would like 10% off, there is a code here, PD10. That's going to be a great seminar if you can attend, so shout out to them. All right, oops, I'll come back to that one in a moment, but all right, so with all the housekeeping out of the way, I am very excited to announce our guest speakers today, where we have both a vet and a phage researcher perspective for this seminar, which is so exciting, a very first for us today. So to start off with, we have Professor Julianne Grose, who is a lab leader at Brigham Young University in Utah. Our lab works on numerous different topics, but one of them being bacterial hosts and their respective phages, and their subsequent use in human and animal pathogens. And for the veterinary perspective, we have Dr. Whitney Greene, who has worked at numerous zoos, aquariums, and animal rehab centres, working with, of course, various animals as well, including elephants and marine animals, which we'll find out, I guess, more today. I guess it's interesting to know that Whitney didn't have much phage experience prior to this case, but was very quickly introduced to the phage world by Stephanie Strathby. So now I'm going to pass over to Julianne and Whitney, who are going to tell us about their story in saving a sea turtle with phage therapy. So thank you, Whitney and Julianne.
Great. Okay.
All right, well, thank you everyone for joining us, and thanks for that lovely introduction. Really happy to be presenting on this with Julianne, and we worked on this case together while I was a veterinarian at Mote Marine Laboratory, and this was for a loggerhead sea turtle who we'll introduce in just a moment, but really interesting case and a really great collaboration between veterinary and research in human medicine. All right, next slide. All right, so first, let me introduce you to Shelly. Shelly is a 40-year-old female loggerhead sea turtle. I came to know Shelly as the new veterinarian at Mote when I started there. And Shelly had SCUD, which is septicemic cutaneous ulcerative disease. So basically a really bad infection in her shell. And though I was not there for the initial presentation of her case, the way it was described to me is that the first lesion started on her carapace, and the carapace is the top part of their shell, so I'll say that a lot. It started as about a dime size region of infected necrotic shell, and it quickly spread to encompass over 75% of her carapace. And she was treated with antibiotics, antifungals. She had surgery. They did lots of cultures and biopsies and every type of topical treatment you can imagine. They got some improvements using manuka honey and a wax that they tried to coat on the shell for a while, along with surgical debridement, but the infection just continued to spread and spread. And the bacteria that was causing this was, not normally a very bad bacteria for sea turtles. It's in their normal flora, but for her, it caused quite severe disease. So when I started, not only did she have this infection, but the real turning point for me that made me start learning about phage therapy, because I really did know nothing about it at the time, was the susceptibility pattern for the bacteria. That's how we were tracking her. It went from being susceptible to a lot of different antibiotics to being multi-drug resistant. And at that point, it was a concern for her. It was a concern for the animal that she shared an enclosure with. And then it was also a concern because we were at a public aquarium, and having a multi-drug resistant bug, didn't know, what that meant in terms of, for the public. So that's when phages came in, and the previous veterinarian had looked into phage therapy, but was really limited based on cost, based on feasibility. It had never been done in a sea turtle, and a lot of other issues. And then he was my mentor, and he's like, why don't you kind of relook into it? And I was like, a bacterial what? I don't even know what that is. And then quickly got roped into it. And we had this great collaboration between Yale and then with Brigham Young with Dr. Julianne. That's kind of how it started. So let's go to the next slide. So this is her shell that we were talking about. So her shell is on the right of the screen. So as you can see, the majority of her carapace, again, the carapace is the top part of her shell, is severely infected. And she has all of these multifocal patches of infected, necrotic, soft tissue, throughout her entire shell. The turtle on the left is her exhibit, her enclosure mate, Montego, who is a healthy turtle, and they've lived together forever. So that was another really interesting, is that why does she have such a severe infection and then, this other turtle didn't? So another component of this project while we were using the phages was looking at some immunology assays over the course of the phage therapy to try and understand more about sea turtle immune system, because we always had a suspicion that Shelly must have had some type of, to be immune compromised in some way. All right, next slide. All right, and I will hand it over to Julianne.
Yeah, so I'm going to really focus on the bacteriophage research. And I want to start off by saying a lot of the phage work was done by Benjamin Chan at Yale University, as Whitney said. So we want to give him credit as well and his lab. But one reason that bacteriophages, as most of you probably already know, one reason that they can be of great use, especially in antibiotic resistant strains, is due to their specificity of action and narrow spectrum of activity. So after being on many antibiotics, many animals can be really deprived of their natural microbiome. And phages can go in and specifically attack just one strain, which we'll talk more and more about. So this tends to make them safer and more tolerable because you're not having different side effects. And of course, there's lots of different ways phages could be administered. And in fact, in this study, it's really interesting because we went for many different routes of administration, IV versus topical, and even into the shell as well. So there were many routes of administration. The effect can be limited to the infection. So as soon as the host for the bacterial host is gone, the phage can often be unstable and be gone. And it's, of course, less expensive than many therapies. So what we designed phage therapy against was a strain of Citrobacter, as Whitney mentioned, that was infecting the sea turtle. And Citrobacter belongs to this family of Enterobacteriaceae bacteria that are really commonly antibiotic resistant. The CDC calls them the carbapenem resistant Enterobacteriaceae. They seem to just acquire antibiotic resistance really easily. And because they're found so commonly in animals, that's where their name comes from, Enterobacteriaceae, meaning they live inside our gut in the enteric bacteria. We see them all over the place, and they're really big causes of antibiotic resistant infections. So what did we look for in phages? Our lab isolated phages, Benjamin Chan labs isolated phages. And I just want to go through some of the critical things we were thinking about at the time. So some of the biggest problems of best practices in phage therapy are first to test for the narrow specificity of the bacterial of the phage. So of course, phages have to bind their bacterial cell at a specific receptor. So they're recognizing their host, and then they inject their DNA, replicate, and eventually lyse the cell. And this initial step in determining their host is usually conferred by something called the tail fiber of the phage, which actually has usually two domains, a structural domain that actually forms part of the fiber, and then a functional domain, which is actually recognizing a receptor on the bacterial cell, and often even sometimes has enzymes for degrading the cell wall and really getting down to the bacteria and the receptor. And that receptor can be lots of different things for different bacteria. It's often protein transporters, it can be the flagella, or even small molecules on the surface of the bacteria. And of course, there are many other things that can confer specificity, but that tail fiber is thought to be the major point of host specificity. But phages also have to be able to lyse the cell. So they have enzymes or proteins called holins that poke holes in the membrane. And then they have lysins that actually break down the cell wall. And these can be grouped into many different categories: glycosidases, amidases, endopeptidases, and that allows the phage to get out of the cell. So the first thing we looked at was the specificity of the bacteria, making sure that it would absolutely infect the nasty bug from Shelly, the Citrobacter from Shelly, but not other bacteria. And let me just point out once again how different strains can be, because in my lab, we had a huge collection of Citrobacter frondae phages that we had previously isolated, and none of them would infect the strain that we got sent from Whitney. So none of our previously isolated phages would infect that strain. So phages are often not only specific for the species that they're infecting, but particular strains that belong to that species. All right, the second thing that we're considering is the transfer of genes to bacteria. So as some of you may know, phages have two possible lyse styles. They have what we call the lytic phase, which is what we've been talking about, and that's where they simply replicate, make a whole bunch of phages and lyse open the cell. And then phages can also go into what we call the lysogenic cycle, where they integrate into the host genome and they just persist in the host cell. So every time the bacterial cell replicates, they get replicated. They can come back out of that lysogenic cycle through induction back into the lytic phase. But we wanted to make sure that we were working with phages that looked like they were just lytic, and that we couldn't see any evidence of the lysogenic cycle. And that's because different phages will spend different amounts of time in these two cycles. Some will be more lytic than lysogenic, and others will be more lysogenic than lytic. And we wanted to make sure we were just using lytic phages for two reasons. One, we wanted the phages to actually kill the bacteria, but then number two, phages actually often contribute to the pathogenicity of bacteria. And this is something a lot of people forget, right? They just think, oh, phages are the answer to disease, but we need to be really careful because phages are often the cause of disease. So this is just a few examples. There's like 30 different examples that I've accumulated over time. And here's just some that you're probably really familiar with. So in each of these cases of these pathogenic bacteria, it's a temperate phage in the genome encoding a toxin that is actually causing the pathogenicity of the bacteria. So this is true for diphtheria, E. coli 01578, the Clostridium botulinum, the botulinum toxin, Streptococcus pyogenes, Vibrio cholerae, and Clostridium tetani. So we have to be really, really careful when choosing phages. We try to fully sequence them and annotate and carefully look at the genes that they carry. we're looking for things like toxins in the examples I just showed you, but phages can also carry biofilm production genes, and biofilm production is also a mode of pathogenicity for many bacteria. The biofilms help protect them from chemicals and allow them to persist in an environment. And then phages have been shown to carry antibiotic resistant genes too. So the last thing we want to do with phage therapy is accidentally make something more pathogenic by using a phage that confers pathogenic genes. So the phages need to be really well characterized. And then the third consideration is bacterial resistance. So just like with an antibiotic, a bacteria can become resistant to the phages that we use in phage therapy. And just like with an antibiotic, we prefer to use phages from different families to overcome this. So the way that I like to explain this is that phages, as shown in the far left in number 1, would normally have a receptor that they're binding, and this is the bacterial cell here, that they're recognizing as their host. But if that bacteria has a mutation and that phage can no longer bind to the host, then it could be resistant to that phage. But the phage in turn, because it's biological, can mutate and change, recognize that new receptor. And the bacteria can do other things like decorate the receptor with small molecules that mask the phage binding, or a protein that masks the phage binding. So there's a war going back and forth between the phage and the bacteria, essentially, of trying to infect the cell and different bacteria becoming resistant to the phage. So what we try to do to win that war with the phage is use families of phages. So we design a cocktail usually with multiple phages that come from different phage families. And when we're talking about a different phage family, this can be done many different ways. And you've probably seen this in several other presentations. We have lots of ways of putting phages into families by nucleotide sequence or by protein sequence. This is actually a phylogenetic tree by the proteomes of different phages, where we were able to group phages into families by the proteins that they carry. And this is actually an even more sensitive way, since phages can not look very similar at all by nucleotide sequence and yet share a lot of the same proteins and hence still be related. So these are the things that we consider in phage therapy, the best practices, but there are still so many other considerations, and most of them are unknowns for us. And I just like to throw them out there so you guys can start thinking about them. Many of you probably already are, and you start designing experiments to try to answer these big questions in phage therapy, which is like, how should we administer phages for different infections, right? Phages have been used as aerosols for infections of the lungs. They've been used topically, they've been used intravenously, right? There's so many different ways that we can use them. And it's a big consideration, like how can we apply them in a clinical case, and what are the best methods? And then each of those methods is going to have its own pros and cons, particularly with how stable the phages are in an animal. And this is something where there's not a lot of work, and we're just starting to see more and more publications on different phages and their stability inside animals. And how are some phages recognized as foreign and attacked possibly by our immune system? Like, what phages does our immune system recognize as foreign and mount an attack against? And what other ones might be left alone because they look similar to the phages already in our gut? So this is just a model of what an animal microbiome might look like. And an animal gut has, thousands and thousands of different bacterial species and probably tens of thousands of phages already. And so somehow our body is recognizing the beneficial bacteria as beneficial and the beneficial phages as beneficial, right? So how can we begin to understand this? Like, what is our body recognizing as foreign versus self? And another big question is phage therapy is, how can we get a better idea of host range? Because when we do host range in the lab, we're just testing the phages against bacteria that we've isolated and that we know of. But the truth is, inside that animal gut is all kinds of bacteria that we haven't fully tested. And really, it's an impossibility to culture all the tens of thousands of different bacteria in an animal gut and test the phage against that. So how can we get better ideas about what the host range will actually be like inside of a living organism? And there's a whole bunch of other big questions. These are just some of them that are always on my mind. But I'm going to show you some of our data now from the sea turtle, which was pretty exciting. So we treated the sea turtle 8 times. As you can see here, the treatments occurred where these dotted lines are over the course of 33 weeks. These are weeks all the way across. And we did monitor the white blood cells, the hematocrit, heterophils, lymphocytes, monocytes, eosinophils, basophils. So basic blood work for the sea turtle. They also did phagocytosis assays where they take the blood serum and look for phagocytic activity, right? So phagocytic activity happens from many of your immune cells, including your macrophages, which when they're activated, they're hyperactivated for phagocytosis, indicating that there's an infection. And this did go down over the course of administering phage therapy. And one really interesting thing that was performed is a look at the stability of phages in the sea turtle, which is rarely done in many cases. So this was really interesting to see that there was a big difference in the stability of phages. Some of our phages just simply wiped out, right? They were pretty much undetectable throughout the study, whereas others actually increased, and by far the majority just stayed pretty constant, with perhaps a big increase at the end. And one thing that already Whitney alluded to is the testing of the bacterial strain over time. And what we see is that even though it was resistant to several antibiotics when we started the treatment, it became sensitive to a majority of them by the end of the treatment. And this has been shown in several phage therapy cases now too. And it's a really important point to note that generally when a bacteria is becoming resistant to an antibiotic, that's a very different pathway than the pathway that the phage is using for infection. And so if it wants to become resistant to a phage, it's still going to stay sensitive to an antibiotic, or vice versa. If it wants to become resistant to an antibiotic, if you're selecting for that resistance, then you're still likely to be sensitive to a phage because they're 2 totally different mechanisms. And now I'll turn it back to Whitney.
Yeah, that was a great detail on everything. This picture right here is just showing the progress of her shell over the course of treatment. As you can see, there was quite significant healing. Now, granted, this picture from February was after a debridement, where we went in and we got rid of a lot of the affected, the infected tissue. But, looking at her every single day, I can tell you that, compared to other treatments, and this, we heard from her, her trainers and caretakers as well, she rebounded quicker with the phage therapy. Her shell hardened up, after those debridements, and the tissue filled in and it was harder overall. The other really cool thing is that, and you can kind of appreciate it from the pictures, is that the pigmentation actually started to come in. So you can see the areas of healing, and you can also see the areas of the new tissue, and that we actually got that pigment. Sea turtles do everything very, very, very slowly. So it's normal for them to take years and years to heal. But, that initial, when they start putting down the fibrin and that initial wound healing, it did seem subjectively faster with the phages compared to previous treatments. and she's doing great. They continue to monitor her, with monthly blood checks to make sure that there's no other involvement in any of her other tissues, and then also, looking at that susceptibility pattern of the Citrobacter to make sure that it remains susceptible to, a lot of the different antibiotics. And I think, though we never fully resolved the Citrobacter, for me, I think the two best points of this whole case, is that one, Shelly's still alive. We were concerned in the very beginning with the multi-drug resistant status and just the severity of this infection. This was an animal welfare concern, and there were discussions about humane euthanasia. And then additionally, the fact that we got it back to a susceptible pattern, that she was able to go back to her enclosure and live a normal sea turtle life. We did document that there were deficiencies in some of her immune cells compared to the control animal that we used. However, that has to be interpreted subjectively because there's just very little known about sea turtle immunology. We gained a lot of information. She's doing really well. And I do really believe that if the infection hadn't been as severe as this was, that it could have completely resolved it. There is one other report out on using phage therapy to treat dysbiosis in green sea turtles in Australia. And I think they had almost a full resolution. The shell and that deep bone infection is really, really hard. But overall, great case. And I think is that our last slide, Julianne?
Yeah, I believe so. Yeah.
Amazing. Thank you so much, Whitney and Julianne. That was an amazing story as always. We do already have some questions in the chat, so I'll just go ahead and read them. But just a reminder to everyone, if you do want to unmute and ask your question rather than typing it in the chat, you can go to, I'm just going to find it myself. I think it's under reactions and the raise hand, and then we can unmute you. But first up, we have a question from Michael. So were the isolated bacterial isolates of the same clone? I think that'd be a question for Julie and me.
Yeah, so you're asking, like, was it from a single colony? I guess I'm a little bit confused about what you're asking. Yeah, when we got isolates from Whitney, we streaked them out and analyzed, and they were clonal. Like we couldn't see any difference between the different colonies, and then we isolated phage against that.
Perfect. Michael, if you wanted to follow up with anything, I'm just checking. Okay, yeah, if there's any follow-up to that, please just ask Michael. The next question we have is from Sabrina. So did you check to see if this multi-drug resistant bacteria was in the feces of the animal?
We did, and it was not. And we checked that periodically throughout the course of this project.
Perfect.
Yeah, so that would suggest that it just got in the wrong place, right? Wrong bacteria in the wrong place.
Yeah. Yeah, super interesting. The next question is from, we have quite a few questions, which is good to see. So question from Henry, is it possible to determine host specificity of tail fibers from sequencing data?
Yes, it is to a certain degree. So that's one thing I'm super interested in. The receptors for most of the tail fibers have just not been determined yet, and it's just highly unstudied. But for the ones that have been well studied, you can completely tell what the host range is going to be from the tail fiber. So that's the good news. Like, I think the more knowledge we'll get, it will be possible to just get a phage sequence and go, oh, that phage is going to be able to infect these hosts. I should say, with one caveat, it is really challenging because I recently put a paper out just a couple of years ago with me and Sherwood Cashins, where we were looking at phages and salmonella, and we were looking at thousands of salmonella strains, right? So same strains of the same species of bacteria, and the different tail fibers of different phages, temperate phages that were living in them, right? And we could tell the species apart. We could tell what the cell wall and the outer proteins would look like based on the phages that were present, which was really, really cool.
That is very interesting. I'm sure there might be some follow-up questions to that as well. Just going through the chat and trying to get through all the questions, but we have a next one from Al. So you mentioned the different routes of administration that you used for the infection. Could you please elaborate on that a little bit, please?
Sure. Yeah. So we used IV. So we did intravenously. We did topically, and we also injected it into her shell. So I guess what a typical treatment session looked like, like when we did that initial debridement in January, we had the first round of phages ready to go. So after we did the debridement, injecting a highly concentrated formulation of the phages into those deep, soft pockets of the shell. Again, the bone infection is really the hardest thing to get. So we wanted to try and get it as deep in as possible. We also drilled holes throughout her carapace, small little, like 1 millimeter holes. And we used a syringe to inject it deep into those drill holes to try and get it as deep in the shell as possible. In addition to the injecting, we, after the debridement, coated her entire carapace with a liquid form of it, like a spray. I think I used a spray bottle and literally just sprayed it all over, trying to allow as much contact time as possible. And then we ended up covering it in Ilex, which is it's like a really amazing Vaseline, that stays on, forever and ever and allows the most contact time possible. We talked about oral formulation, but we all kind of agreed that just due to where the infection was and how deep it was, that probably was not the best way to use our resources. And then we had an intravenous formulation as well that we used.
Perfect. Thank you, Whitney.
And then there's also, we had a high concentration in the tank water too, which was really cool. So we had to figure out, that was a huge learning thing, is figuring out all of the water chemistry to keep the filtration off and just have a really high concentration of them in her enclosure. Yeah.
Perfect. Yeah. And I guess it's a bit of a follow-up to that. With the phage stability graph that you showed, Julianne, was that from the collection of blood samples from Shelly, the turtle, yeah. Was it? Okay. And do you think the ones that were completely undetectable, were they still able to lyse the host? So was it that they were undetectable because they didn't have a host, or is it because you think that they're unstable upon entry?
They're still able to lyse the host, so they would have been unstable upon entry. Yeah.
Right. Super interesting. I think
Great
that's
question.
super interesting.
I should have clarified that. Yeah.
No, that's great. So Michael's asked, did you get a whole genome sequence of the isolates, or the isolate that you had from Shelly?
Of the bacterial isolates. We haven't yet. I don't know if Whitney has. Have you sequenced the whole genome of the bacteria, Whitney?
I think they were going to, but I changed institutions, so I don't know what has come from that.
Yeah.
But that was in the plan.
Perfect. So a question from Jean Paul. Did you use a combination of phages and antibiotics together? Thought I saw that some antibiotics like penicillin show some in vitro activity. Oh, sorry, not penicillin.
Yes, we did use, we used several antibiotics throughout the course of this, and we chose those antibiotics based on the culture results that we were getting. But it seems like the antibiotics worked really well, with the phages. But again, those were, it's getting to the actual site, so I do think they helped, but we never were able to clear it. But we did mostly oral, and then we did IV antibiotics for a while as well.
Okay. The next question we have, how do you check if the bacterial isolates are clonal?
Yeah, that's a great question. So really it just a few different point mutations can lead to big differences between different strains is the truth. And so it's hard to even sequence to show that things are not, that things are clonal. You would have to get the exact entire full genome sequences of lots of colonies off the plate to show that it's clonal. An easier way for phage researchers is to just choose, look on the plate the way that we do it. We streak it out, we look at the plate, we see, does there look like there's any obvious differences in the colonies? And if there's not, then we go ahead and choose 1 or 2 colonies and we treat them as separate isolates and we isolate phages against them. And then we test and we see, are all the colonies susceptible to those phages, right? Because if they're all susceptible, then if it's not clonal, it's at least very close to clonal. So.
Yeah, and you performed that, did you say? I think that
Yeah.
was a follow-up question to that
Yeah.
as well, yeah. And I guess there's a suggestion in the chat just to maybe anyone who's interested as well. So you can do in vitro studies to look at the synergy between phages and antibiotics used. So a good point from Jean-Paul.
Absolutely.
Yeah. Is there any other questions? You're welcome to unmute or put them in the chat. I guess I have just a quick question. So just looking at the picture again, it looks like the last phage administration was around January 2020. So we're almost 2 years away, like 2 years since that. Has there been any other phage administration needed, or like I know you said that she's mostly recovered, still got the Citrobacter, but has there been any other major interventions between then, or she's pretty much fully okay just to live out her life as is?
From what I know, she's doing great. They do periodic. I think they've gone as frequently as once a month to about every 4 months, just looking at her blood work and then looking at cultures. But as far as I know, there has not been any need for, anything more. So her shell's never going to look normal. So that is, understood, but she's doing good.
So that's amazing. And she was able to be reunited with her her mate that you mentioned at the start that wasn't infected at all.
Yes, her and Montego are living their best life.
Amazing. We joked about at the start that we should have like a live cam to the sea turtle, but that would have been funny. Oh, there's another question from Michael in the chat. Was the IV treatment and topical treatment applied at the same time? If they were given at different times, did you see a difference in healing following each of the applications?
Some of the times they were at the same time and sometimes it was different. So unfortunately, there was really no real pattern to that. We had to be really coordinated, in terms of receiving the phages and handling Shelly and trying to, optimize her welfare as much as possible. So it was a bit random.
Yeah.
So I didn't really appreciate any, correlation with one or the other.
Yeah, perfect. Any other questions that people have? And I guess for some more details, we did link the paper that was I guess recently published on this story as well, so there might be some more details in there as well. Sabrina, I may have missed this, but antibiotics added to the water, were antibiotics added to the water to prevent infections in these habitats? So maybe routinely or throughout as well?
No, in general, not with sea turtles. I mean, you can do antibiotic therapies for topical infections, but we never did with her, just due to the extent and the severity. We did we played with the salinity a little bit, prior to the initiation of the phages, but decreasing her salinity. But yeah, never with antibiotics.
And I remember there being something about the water changes of because I can't remember the how often the water's usually changed for the sea turtle enclosures, but you had to I guess change that when you added phages. Was that
right?
Something that I remembered?
Yeah, they're and I'm not the person to speak on that. It's very water quality and water science is very complicated. But we had a whole team of people and we had to do lots of calculations and lots of plans about. There's just a very delicate balance between the biological filtration and keeping everything nice and clean, and how we had to turn basically all of that off, all of the ozone and everything had to go off, for the phages, otherwise it was going to impede that, so how long could we basically have everything off without worrying about the water quality getting really gross, for lack of a better word. and then disposing of it appropriately.
Yep. And I guess maybe this is a bit of a broad question, but what was the main takeaway and sticking point for getting the phages into the sea turtle? It's two different angles there, but main takeaway and main sticking point.
For me, it was a last ditch to try and get her as healthy as we could. It really was between, looking into this, phage therapy versus potentially considering, quality of life monitoring, with the end result being euthanasia. And it was something that was completely unknown to me, so I think that it's an amazing treatment modality. There are so many cases that I'm always like, this could benefit from phages and this could benefit from phages. So it really opened the door to me for that. And then the other really cool thing, was just learning more about their immunology and how that plays with overall health and the way they're healing and immunology and everything.
Perfect. Thank you, Whitney. Anything you wanted to add to that, Julianne?
No, I agree. There's just so much unknown. As I said, we don't really understand, how our phages treated different by our immune system if we put the phages in the turtle shell versus intravenous versus topical. What different immune interactions are there, and how is the immune system recognizing the phages as foreign or self? And there's just so many unanswered questions.
Yeah, for sure. I think everyone in this audience can appreciate that as well. Yeah. Well, I think in the interest of time, I might wrap it up there. But thank you so much, Julianne and Whitney, for making time to come and talk about this story. It's an absolutely amazing story that I love from the very first time I heard it, and I think everyone would have loved hearing more about it today. So thank you again. You're more than welcome. I know you're both so super busy, so you're more than welcome to stick around for the casual networking, or you can jump off and get on with your day.